When you look at a vibrant forest canopy, the volume of overlapping biological life is immense. Measuring it requires isolating the chaos. Ecologists start by defining alpha diversity. This is the specific measurement of local diversity within one single site or habitat. Alpha diversity asks how diverse one specific pond or single forest patch is. It strictly isolates the local environment, stripping away beta diversity, which compares turnover between different habitats, and gamma diversity, which calculates the total regional landscape. To measure this isolated site, the most instinctive method is to calculate species richness, represented by the variable S. This is simply counting the total number of distinct species present in the habitat. While richness is the starting line for any ecological survey, it contains a significant mathematical blind spot. It ignores evenness. Evenness is the measure of how the individual organisms are distributed among those species. This side-by-side bar chart illustrates the discrepancy. On the left, 10 species are perfectly equal. On the right, a single species dominates with 1,000 individuals, while the other 9 have exactly 1. Under a simple richness count, both score 10. They look identical. The extreme abundance ratio of 1,000 to 1 masks the vulnerability of the rare species. One localized disease or weather event could eliminate 9 entire populations in the second habitat, making the simple richness count an insufficient metric for assessing the health of the community. Local biodiversity measurements require complex mathematical models that force richness and evenness to interact. Before we can evaluate evenness, we have to correct a flaw in how data is collected. This is the sampling bias imperative. Researchers who capture more individual organisms will naturally find more distinct species. This equation is Margolef's richness index. It corrects sampling bias. In the numerator, we take total species count, S, and subtract 1, establishing a 0 baseline. Let's plug in 10. In the denominator, we divide by the natural logarithm of n, the total individuals sampled. With a baseline of 100 individuals, the calculation outputs a corrected index of 1.954. Once the sample size is stabilized, ecologists deploy tools that actively measure evenness. The most common is the Shannon-Wiener Diversity Index. This data matrix breaks down the index. Shannon relies on calculating the proportional abundance of each individual species relative to the whole community. If species A has 50 individuals out of 100, its proportion is 0.50. The core mathematical mechanism involves multiplying that proportion by its natural log. Because proportional abundance is a decimal between 0 and 1, its natural log is negative. For species C, with a proportion of 0.20, the natural log is negative 1.609. This logarithmic function amplifies the signal of rare species because smaller fractions yield disproportionately large negative logs. We take the summation of the column, which gives us negative 1.030, and apply the formula's negative multiplier to reach a final positive output of 1.030. As an analytical scale, a Shannon score under 1.5 indicates low diversity. Scores over 3.5 indicate high diversity. This index has a mathematical ceiling. Maximum diversity is achieved only when every distinct species in the habitat has the exact same population size. This maximum is calculated as the natural log of the total species count. The Shannon index is the preferred lens when researchers must ensure that low-abundance, rare species are not mathematically overshadowed by larger populations. While Shannon emphasizes the rare, Simpson's index operates on an opposing mathematical logic based on strict probability. It asks, if you pull 2 individuals at random from a habitat, what is the probability that they belong to the same species? To find this probability, Simpson's formula squares the proportional abundance of each species. Squaring decimals significantly affects the results. Large proportions remain relatively high. 0.60 squares to 0.360. However, small proportions diminish. A 15% population share drops to 0.023 in the final sum. The output of this sum is called lambda, representing dominance. A higher number indicates that a few species are dominating the total population. To measure diversity rather than dominance, ecologists use Simpson's Index of Diversity. By subtracting the dominance score from 1, the metric is flipped. Researchers can also apply Simpson's reciprocal index by dividing 1 by the dominance score. This scales the output up to the total number of species. Ecologists use Simpson's index to prioritize the impact of highly abundant species that dictate the habitat's resources. Researchers often need to strip away the total species count and look exclusively at the mechanics of population distribution. This gauge tracks Peelew's evenness, a tool for isolating pure equality in a habitat. It takes a community's Shannon score and divides it by the theoretical maximum evenness. The output is a ratio, where 1.0 represents perfect mathematical equality among all species. Here, our sample hits 0.937. To shift to the opposite extreme, ecologists isolate dominance using the Berger-Parker Dominance Index. Berger-Parker is calculated by taking the population of the single most abundant species and dividing it by the total community population. Extracting these extremes provides a clear metric for resource distribution. A dropping Peelew score, or a spiking Berger-Parker score, signals that a single species is outcompeting the rest of the community. Observed data is rarely the complete reality. In most field studies, undetected species are present but remain uncounted. To compare sites fairly, ecologists use rarefaction. This process mathematically subsamples a larger dataset down to the size of a smaller one, ensuring two different environments are evaluated on the same scale. To project the true species richness of a habitat, ecologists apply the Chow-1 estimator. It uses the frequency of rare sightings to estimate what was missed. It tracks singletons, species seen exactly once, and doubletons, species seen exactly twice. If a researcher records 20 total observed species, with 6 singletons and 3 doubletons, we plug those values into the formula. The logic is that a high number of distinct species seen only once implies that additional species exist in the habitat that were not seen at all. The equation squares the singletons and divides by the doubletons, adding 6 estimated species to our total. This turns our 20 observed species into a projection of 26 true species. Missing data is a mathematical variable that can be solved to reveal the actual richness of a living community. Ecologists select specific mathematical lenses based on the biological realities they need to measure. This analytical matrix dictates those choices, providing a framework for fieldwork and ecological analysis. The first rule of analysis is Shannon shares with rare. Shannon is used when the impact and presence of low-abundant species are the primary focus. The second rule is Simpson sides with dominant. Simpson is chosen to track the influence of abundant species that characterize the habitat. Finally, Peelew's evenness measures distribution equality, while Berger-Parker isolates the most abundant player in the system. Alpha diversity serves as a mathematical audit of a community. It transforms a simple list of species into a weighted assessment of how individuals are partitioned across the ecosystem.